today for this President's Cup and Thematic session number 1. So a few things, and actually start off with one reminder. Would you mind, if you haven't already, if you could please vote on our election for the executive board? If you haven't done so already, that would be great. Today we'll have 4 presenters, and logistics of this thematic session number 1 will be each presenter will be presenting for 8 minutes. They will not have questions right after their sessions. However, if you do have questions, please just put it in the chat. And then depending on time and when we're done, I'll be asking the questions at the end. And once again, if you could please kind of direct on where the questions are going to, so I can direct the questions to the right person here. So with that, I'm going to introduce our first presenter here, and I'll stop sharing, but Donna will be first here.
Okay. Hi, everyone. So my presentation is on the difference in steady state chemoreflex drive between groups with and without a patent foraminal Okay, sorry, it's not letting me advance forward. Okay, there we go. So currently, to investigate the ventilatory response to high altitude either requires steady state or transient hypoxia testing or invasive blood gas measures. In the field, these techniques have many caveats, including the consideration for safety, portability, and consistency. In response to these challenges with fieldwork, another method was developed called the steady state chemoreflex drive, abbreviated SSCD. This method investigates the overall ventilatory response utilizing indirect measures of minute ventilation, end-tidal carbon dioxide, and peripheral oxygen saturation. Specifically on the left graph, you can see that the partial pressure of oxygen in the arterial blood has a hyperbolic relationship with ventilation. The SSCD takes advantage of the inverse linear relationship between arterial oxygen saturation and ventilation. Additionally, the SSCD also uses the indirect linear relationship between the partial pressure of carbon dioxide in the arterial blood and ventilation. So through their accumulative effect on resting ventilation, the stimulus index can be defined as the end-tidal carbon dioxide over the peripheral oxygen saturation to calculate the stimulus index. And then with ventilation or minute ventilation and the stimulus index, the SSCD can be calculated. So our purpose was to assess whether this new, simple, non-invasive approach, the SSCD, could show differences in the overall ventilatory response between groups with and without a patent foraminal valley. So patent foraminal valley, which I'll refer to as a PFO, is a shunt located between the right and left atrium. So in the picture, the right atrium is the top blue circle, while the left atrium is the top red circle. And this white arrow is where the PFO would be located. In utero, this shunt supports fetal development and closes at birth. In approximately 30% of the population, our lab has found that this shunt remains open. So ventilatory acclimatization is the progressive increase in ventilation in response to chronic hypoxia. So using the arterial or the partial pressure of carbon dioxide in the arterial blood as an index of ventilation, you can see on the graph the hypoxic ventilatory response. If we focus in on just this dark gray area, this is the ventilatory acclimatization phase of the hypoxic ventilatory response. And so it demonstrates that as hours to days increase, the partial pressure of carbon dioxide slowly decreases. And so to combine PFO with ventilatory acclimatization, previous studies in our lab have shown subjects with a PFO, which I'll refer to as PFO positive, versus subjects without a PFO, which I'll refer to as PFO negative, have a reduced degree of ventilatory acclimatization. So you can see in the graph that the open circles represent the PFO positive subjects, while the closed circles represent the PFO negative subjects. For a change in ventilation over the change in saturation, you can see that ALT 16, it stands for altitude day 16, that the PFO negative group were significantly different from the PFO positive group, denoted by the double cross. Which means that for the PFO negative group, they had a higher ventilatory drive than the PFO positive group. This could be problematic at altitude, as a reduced degree of ventilation could be, for PFO positive subjects, could make them more susceptible to high altitude illnesses. And so we performed a retrospective analysis from the high altitude expedition called Altitudomics that took place in Bolivia in 2012. Specifically, 21 participants, 11 of whom were PFO positive and 10 of whom were PFO negative. Data were collected from sea level, day 1 at 5,260 meters, and day 16 at 5,260 meters. Specifically, at these time points, minute ventilation, end tidal carbon dioxide, and peripheral oxygen saturation were used to investigate whether the SSCD, which again takes the end tidal carbon dioxide over the peripheral oxygen saturation to define the stimulus index, and then takes minute ventilation over that stimulus index to calculate the SSCD. And we wanted to see whether the SSCD was able to show differences between PFO positive and PFO negative groups. And so what we found was, in comparing the SSCD between PFO positive and PFO negative individuals, that there was no difference in the SSCD between groups, with a p-value of 0.3871. And so in the left graph in pink, you can see the PFO negative group, and then in black is the PFO positive group. On the y-axis is the SSCD, and on the x-axis are our time point sea level, ALT1 and ALT16. And so you can see here that even though the PFO positive subjects were higher than the PFO negative subjects were higher than the PFO positive subjects, there was no differences between groups. Interestingly though, in the multiple comparisons test, for the p-values did become closer and significant between the PFO positive and PFO negative group as time at altitude increased. Additionally, there was also a significant effect of time, with a p-value less than 0.001. And so on the right is a graph showing alveolar ventilation from the same data set. And so again, you can see on the x-axis are the time points sea level, ALT1 and ALT16. On the y-axis is the partial pressure of oxygen. In the open circles are the PFO positive subjects, while the closed circles are the PFO negative subjects. And so at altitude day 16, the data showed that the partial pressure of oxygen in the alveolus were significantly different between groups, as shown by the double cross. And then additionally, with the alveolar data, the asterisk and the single cross show that in comparison to the previous altitude, there was a significant difference. And so we demonstrated that although the SSCD between groups did become closer to significant as altitude increased, the SSCD did not show significant differences between the PFO positive and PFO negative individuals, despite alveolar ventilatory data showing that there were differences between groups. We also demonstrated that as the SSCD did show a main effect of time, which was consistent with the alveolar ventilatory data. And so again, since our purpose was to assess whether the SSCD would show differences in the overall ventilatory response between PFO positive and PFO negative individuals, our conclusions were that the SSCD may not be sensitive enough to show differences between groups, but can accurately determine the overall ventilatory response to altitude. And so I'd just like to thank the Altitudomics investigators, the funding agencies for allowing me to use their data, as well as Dr. Levering in the lab for helping me with this experiment, and ACSM for allowing me to present.
Thank you. Okay, Tyler is up next here.
There we go. My name is Tyler Kelly. I am a PhD student in the Cardiovascular and Pulmonary Physiology Lab at the University of Oregon. And I'm here to talk about the high prevalence of Patrick-Freeman Valley found in recreational to elite breath hold divers. So as previously covered, the Patrick-Freeman Valley is a normal aspect of the fetal heart. It is a source of endocardiac right to left shunt. And in the fetal development, it allows the fetal circulation to bypass the pulmonary circuit. In around 1/3 of the population, that Freeman Valley fails to completely close. So that process occurs during the first few moments to few months of life. As an infant takes its first few breaths, left atrial pressure increases relative to right atrial pressure. And that causes kind of tissue flaps that form the Freeman Valley to press against each other and close that tunnel. And over the next few months, that tunnel those flaps rather endothelialize, and that Patrick-Freeman seals. In adults with the Patrick-Freeman Valley, there's an association with migraine, transient ischemic attacks, strokes, as well as decompression sickness with diving. And so it can have some important pathophysiological consequences that we've been looking at. What we did here was we look at some apneic divers, so individuals who compete either for depth or breath hold times, and we evaluated them for PFO. The apneic divers were recruited from international training camps in Sovtak, Croatia, as well as Split, Croatia. That gave us a total of 36 divers, 9 of which were female. Our control subjects were recruited from the general population of Split as well as Eugene, and that gave us a total of 17 control subjects, of which 3 were female. The PFO screening was conducted with transthoracic saline contrast echocardiography, and so that involves the agitation of saline and injection into the antecubital vein while monitoring the heart for the appearance of those small air bubbles in the saline contrast. As you can kind of see in this image here, this top left image is the injection of the microbubbles. And so you can see that when they arrive in the right atrium, they're very obvious to have that nice opaque cloud effect. And as you can see, particularly in the lower right frame there, when there is a Freeman Valley, we can see those small agitated saline contrast bubbles pass through the PFO and show up in the left atrium. And when we see that happen, that tells us that a PFO is present. In addition to the PFO screening, we also took anthropometric measurements and did some basic spirometry. Prevalence of PFO was compared between divers and controls using the chi-squared test, and anthropometric and spirometric measurements were compared using Student's t-test. What we found is that apneic divers had a significantly higher prevalence of PFO. Roughly half of the PFO subjects had, or rather half the diver subjects had a PFO, compared to our control subjects where it was around a quarter had a PFO. We also found that divers were taller than controls and had a lower BMI. However, outside of that, the lung function between our divers and our controls was very similar, as was cardiac function and structure. We have considered a couple of reasons as why there might be a higher incidence of PFO in apneic divers. One suggestion is that there is an increase in hyperbaric, or an increase in pulmonary vascular pressure associated with hyperbaric stress from repeated diving, as well as centralization of blood into the thorax associated with mammalian dive reflex. That combination may cause a back pressure buildup through the pulmonary circuit, so an increase in right ventricular pressure, an increase in right ventricular afterload, in turn causing an increase in right atrial pressure. And as I mentioned earlier, when right atrial pressure exceeds left atrial pressure, that's when we can see flow through that PFO. So it could be that that increase in right atrial pressure associated with diving basically blows open an incompletely sealed PFO, making it more apparent to saline contrast, or making a PFO exist where one is under normal conditions. also possible that this may be some protection against pulmonary barotrauma. There also may be a self-selection influence with a high incidence of PFO in apneic divers. As I just mentioned, there is an increase in pulmonary pressure with the thoracic concentration of, or I shouldn't say concentration, but thoracic movement of blood from the periphery to central compartment. And it could be that the PFO serves as basically a pressure relief valve. And so that these divers who are able to achieve these very long breath hold times or very deep dives are using the PFO to limit the increase in pulmonary pressure associated with that diving, which also may be a protection against pulmonary barotrauma associated with diving-induced hyperbaria. In conclusion, we have a significantly higher prevalence of PFO in our recreational to elite apneic divers. That may be a result of increased right ventricular afterload associated with lung compression and blood centralization. It may also be a mitigating factor to pulmonary barotrauma and act as a self-selection wherein the divers who are successful with apneic diving have the presence of a PFO as a protective. These studies were funded through the Burroughs Wellcome Fund and the Fulbright Scholars Grant, both to Andy Lovering. Thank you for your time with today's presentation.
Thank you, Tyler. Just as a reminder for everybody too, if you do have questions, please make sure to utilize the chat. But thank you very much, Tyler. All right, next up, Carly.
Sorry about that. I had to change up the order. Okay, so the title of my talk is Influence of a Patent Foramen Ovale on Heart Rate Core Temperature Relationship at Rest and During Exercise in Young Healthy Men. So we know that we'll have increases in core temperature, whether we're exposed to passive or active heat stress. And in a given individual, as their core temperature increases, their heart rate is also going to increase. And that's due to two mechanisms. First, we have the effect of temperature on our SA node. So our SA node is very sensitive to changes in temperature. So as it heats up, it's going to increase its firing rate and increase heart rate through that mechanism. And then we also have the activation of the sympathetic nervous system. So we'll have an increase in norepinephrine release, binding to our beta-1 receptors and increasing heart rate. We also know that there's biological variability in core temperature values. So if we bring two people into the lab, chances are they're not going to have the same exact temperature on that day. Somebody could be a little cooler, somebody else could be a little warmer. So our question is among individuals, are these differences in core temperature associated with differences in heart rate at rest and during exercise? Our lab is very interested in studying people with a patent foramen ovale, or a PFO. And this is an intracardiac shunt. And in these people, they have the blood that flows directly from the right atrium to the left atrium. And this blood bypasses the pulmonary circulation. So previous studies out of our lab have shown that PFO positive men have a higher core temperature at rest and during exercise, specifically a VO2 max test. And that difference is about 0.4 degrees Celsius. So we were wondering if this higher core temperature could influence their heart rate responses and if there's a potential for an altered cardiovascular and thermoregulatory relationship in PFO positive subjects. So the purpose of our study was to test whether one, there's a relationship between inter-individual values for heart rate and core temp at rest and during exercise, and whether the presence of a PFO alters that inter-individual heart rate and core temp relationship. So is there a correlation between heart rate and core temp among individuals at rest and during exercise, and does a PFO influence that relationship? We had 30 young healthy men participate in this study, and on their very first day, they came into the lab and they underwent ultrasound screening to determine whether or not they had a PFO. And we ended up enrolling 15 PFO positive and 15 PFO negative subjects. After their initial screening day, they came back to the lab 2 more times to do 2 different exercise tests. The first test, they did a VO2 max, and that was to determine maximal aerobic capacity, but also to determine their workloads for their next visit where they'd complete a graded exercise protocol. That protocol consisted of 4 2.5-minute long stages where they cycled at 25%, 50%, 75%, and 90% of VO2 max. We measured heart rate and core temp continuously throughout this protocol, and it's important to note that all these tests were completed in a thermoneutral, just regular lab environment, so at around 21°C. This was a retrospective data analysis, but we looked at data pre-exercise at 50% and 90% of VO2 max, and we performed linear regressions. So the very first analysis we wanted to do was we wanted to see whether or not there is a relationship between inter-individual values for core temp and heart rate and our whole group data at rest. So you can see that we found there was a significant and positive relationship between core temp and heart rate, and those who came to the lab with core temps around 36 degrees, they had the lowest heart rates, whereas those who came in a little warmer at 37 degrees, they had the higher heart rates. So now we established that this relationship existed in our whole group data. What happens when we divide them out to PFO positive and PFO negative? So looking at this top panel, this is our PFO negative data. You can see there's absolutely no relationship between these two variables at rest. This line's pretty much flat. But you can see if we look at our PFO positive data, they also have a significant positive relationship. So the cooler people had the lower heart rates. Those who were a little warmer had higher heart rates. So we have our pre-exercise data. Then we wanted to see what would happen if we asked them to exercise. So then we had them exercise at 50% of VO2 max, and still in our whole group data, we have this positive and significant relationship between core temp and heart rate. We divide our groups out. We still have a relationship, but this time it's in our PFO negative subjects. So those who were cooler had lower heart rates, those who were warmer had higher heart rates, but in our PFO positive group, there was no relationship between heart rate and core temp anymore. It's important to note that these subjects were all at the same relative intensity, so they're all at 50% of VO2 max, but there was also no difference between absolute workloads between these two groups. So we have this 50% of VO2 max when we ask them to work a little bit harder. There's still that positive and significant relationship in our whole group data at 90% of VO2 max. And we see something similar that we saw at 50% of VO2 max. So we have that positive and significant relationship between core temp and heart rate in our PFO negative group, but there's still no relationship and more variability between core temp and heart rate in our PFO positive group. So in conclusion, there are differences in the thermoregulatory and cardiovascular relationships between PFO positive and PFO negative subjects, both at rest and during exercise. When we were talking about some possible reasons for this increased variability in PFO positive subjects, there's some data coming out of our lab that's showing that PFO positive subjects have higher levels of inflammation. So that could be leading to higher core temperature values, almost like a mini fever, but also leading to increased variability in these heart rate responses once people start exercising. There could also be the influence of duration of exercise stage. So they're all about 2.5 minutes long. So maybe if they had been able to reach steady state, we would have seen some different values. So future studies should examine the influence of a PFO on the heart rate core temperature relationship during a longer duration exercise trial. And that's it.
Great. Thank you, Carly. Good job. And then Caitlin will be last but not least.
Okay, here we go. Okay, perfect. So today I'm going to be talking about the role of circulating inflammation in regulating pulmonary pressure at altitude. A little bit of background information about some of the physiology I'll be talking about. The first important factor is hypoxic pulmonary vasoconstriction, which, as you can see from this drawing, this zoomed in alveoli, that we have an alveolus here with a PO2 of 100 millimeters of mercury and normal blood flow going to that alveolus. If we then look at this bottom right diagram, this is a hypoxic alveolus with a PO2 of 50 millimeters of mercury. And you can see by these arrows that this response is then going to constrict this blood vessel in order to divert blood away from this hypoxic alveolus and towards more well-ventilated alveoli in the lungs. It's important to note that this doesn't require any input from the central nervous system. The main stimulus for this to occur is the alveolar hypoxia. And this becomes significant when we talk about altitude because we do get global lung hypoxia that's going to cause global vasoconstriction. For this study, we tried to look at some of the modulators of this response. So even though this is the primary stimulus for this is alveolar hypoxia, there are some pretty key regulators, namely nitric oxide and endothelin-1, which can regulate this response. Nitric oxide is a potent vasodilator and endothelin-1 is a potent vasoconstrictor. Some of the factors that we wanted to investigate with this experiment was the role of circulating systemic inflammation, as well as a patent foramen ovale on this response. So like some of the other presenters have talked about, what is a patent foramen ovale? It is a right to left shunt. So it allows blood to flow from the right atrium into the left atrium and bypass pulmonary circulation. Some of the consequences of having a PFO are having lower arterial partial pressures of oxygen, as well as worse pulmonary gas exchange efficiency. There have also been studies that have shown increased incidences of HAPE or high altitude pulmonary edema in those that have a PFO. HAPE becomes particularly important in the context of this study because those that develop HAPE are found to have exaggerated pulmonary pressure responses to altitude. The last important bit of background information is the inflammatory response to hypoxia. And so as you can see again from this these zoomed in alveoli, we do have resident macrophages and neutrophils and other immune cells present in our lungs. And those immune cells, when they receive a certain stimuli, will release proteins called cytokines. These cytokines are the main proteins that regulate our immune response. And they have a ton of different downstream targets. So namely, they'll cause infiltration of immune cells that can help amplify the inflammatory response. They can also affect epithelial cells and smooth muscle cells. So taking all of that, the purpose of this study was to determine whether the presence of a PFO or circulating inflammatory cytokines would affect the pulmonary pressure responses during 10 hours of normobaric hypoxia. So in order to do this, we recruited 36 subjects. Those subjects were matched for PFO status as well as sex. And so there were 18 PFO negative and 18 PFO positive. And within each of those PFO groups, there were 9 males and 9 females. We screened for PFO on a separate day with ultrasound. And on our main study day, which is pictured here, we had subjects come into the Evanuk environmental chamber, which is a normobaric hypoxia chamber. And we had subjects breathe 11.5% oxygen for 10 hours. During that time, we took pulmonary pressure and cardiac output measures via ultrasound and then used that to calculate total pulmonary resistance. We also took blood samples at the beginning of their stay in the chamber and after 10 hours. Those venous blood samples were then analyzed via flow cytometry for the presence of 13 different inflammatory markers. 12 of them were pro-inflammatory and 1 was anti-inflammatory. So jumping into some of our results, I will start with the pulmonary pressure data. So as you can see from this graph here, we have our pulmonary artery systolic pressure on our y-axis and our time on our x-axis. And as you can see, there is a significant increase over time in pulmonary pressure the longer they were in the chamber. And all of the symbols you can see down here. The pulmonary pressure did seem to plateau right around 7 hours. We also looked then at the 2 factors that could be affecting pulmonary pressure, namely cardiac output and total pulmonary resistance, which is an indicator of vasoconstriction. And you can see a similar trend with cardiac output where there's an initial increase within the first 30 minutes, and then a slight plateau until right around 7 hours when it seems to increase again. There's a similar trend with our total pulmonary resistance as well. Interestingly, though, we don't really see a response until right around 4 hours, indicating that there's likely not much vasoconstriction happening until that time, and that vasoconstriction isn't causing this initial increase in pulmonary pressure. That initial increase is likely due to the cardiac output. Breaking it down then, whether there were any differences between PFO negative and PFO positive subjects, you can see that there's a main effect over time. Both our PFO negative and PFO positive subjects significantly increased their pulmonary pressure over the 10-hour time period, but there were no differences between our PFO groups. Similarly, with our cardiac output data, you see a very similar response. that both groups significantly increase over time, but there's no differences between groups. And once again, same with our total pulmonary resistance, that you see significance over time, but not between PFO negative and PFO positive. Jumping into our inflammatory data, this is showing the main effect of hypoxia with all of the 13 cytokines that we looked at. And as you can see, all 13 significantly changed. MCP1 right here and interferon gamma significantly decreased, and all other proteins significantly increased. Knowing that both our pulmonary pressure and our cytokines increased over time, we then wanted to see if there were any correlations between the two. And as you can see from these graphs, there were no significant associations between the change in any given cytokine and the change in pulmonary pressure. These are just a few representative graphs, but there's similar pattern with all 13 of the proteins that we analyzed. We also wanted to split up based on PFO status to see if there was any obvious grouping. So there's PFO positive in the open blue circles, PFO negative in the closed red. And you can see that there's no significant grouping with the PFO status either. However, when we do look at a multiple regression analysis of these cytokines, we did find a significant regression analysis to predict the change in pulmonary pressure. So you can see here that the change in IL-18, the change in IL-23, and the change in IL-33 taken together significantly predict the change in our pulmonary pressure. The R-squared for this regression analysis is about 0.37. So this indicates that about 37% of the change in pulmonary pressure can be explained by the change in these three cytokines, which might not seem like a lot, but like I noted at the beginning, the main stimulus for this response is the alveolar hypoxia. And so getting this significant R-squared value and this significant regression does show that there likely is an effect of inflammation on regulating pulmonary pressure. So in conclusion, we found that a PFO does not significantly contribute to the change in pulmonary pressure, and that's likely due to insignificant differences in alveolar hypoxia. Additionally, no single inflammatory marker contributes significantly to the change in pulmonary pressure, but inflammation as a whole from that multiple regression analysis may affect the degree of pulmonary pressure at altitude. So I'd like to give a huge thank you to everyone in Dr. Lovering's Cardiopulmonary and Respiratory Physiology Lab, past and current. This was a huge project and took an incredible amount of work, and would like to extend gratitude towards all of the subjects for sitting in a chamber for 10 hours for us. This research was funded by the Department of Defense, as well as the Eugene and Clarissa Evanuk Memorial Graduate Fellowship. And that is all I have.
Thank you all very much. I learned a lot about PFO. Thank you. Thank you. So I have some questions here. Once again, if there are further questions that you might have, even with some of the questions that I ask, if there's specific follow-up, especially the people that wrote it, please let me know. The first one that I have here is for Donna. So why the interest in individuals with PFO and altitude in the overall data set?
Yeah, so a third of the population has a PFO. And so I showed that data that had like the change in saturation over the change, or the change in ventilation over the change in saturation. And that data was from Altitudeomics, and they had found that people had, with people who had the PFO, they had that bun and ventilatory response and increased incidence of acute mountain sickness.
Nice, thank you, thank you. The next one I have is for Tyler. So where did the idea come from to look into the incidence of PFO in extreme divers?
Well, previous data from the lab has shown that individuals with a PFO have a blunted hypoxic ventilatory acclimatization. And so we wanted to use forms of hypoxia in order to look at that a little bit more. And in terms of groups that regularly are subjected to a chronic intermittent hypoxia, you're kind of limited to individuals with sleep apneas or these breath hold divers. And with someone with sleep apnea, they're asleep, and there are a whole bunch of physiological alterations that go along with that. Where with breath hold divers, they're awake. when they're experiencing this intermittent hypoxia. And so they're a good population to study if you're looking for the effects of reversible forms of hypoxia.
Mhm. Yeah, all right. Thank you. Thank you. The next one I have is actually for Tyler as well. So do you consider the one sample potentially being sampling bias? Do you think these, as a follow-up to that, do you think these results are replicable?
If they're referring to where we sampled from, that is the areas around South and Split, Croatia. Those were international training camps. And so the individuals that we were sampling there, who were there for training camps, were coming from around the world. So we had a fairly diverse ethnic makeup there. In addition, we didn't just recruit elite divers, we also recruited a variety of like competitive spearfishers, things like that. So we not only had a large mix in terms of ethnic backgrounds, but also in their duration and experience with apneic diving. So on the one hand, we had the breath hold divers who are going for depth, where they're probably diving for several minutes, or if they're going for static apnea, they're going for maybe up to around 10 minutes, a little short of that, but for very long times. So good range there, good range in ethnic backgrounds. And in terms of our control subjects, sampling both from Split and Eugene, we have a lot of geographic diversity there, which is also going to be reflected in a lot of diversity in our subjects. And so based off that, I feel like the results we found are going to be pretty highly generalizable.
Nice. Thank you. Thank you. The next one's for Carly. With PFO, is it yes/no, or with yes, are there degrees of severity within PFO?
It's graded on a scale from 0 to 5, and it's based on.
So James, do you have it or you don't?
Sorry? Yeah, just the grading scale. And then did you take into account the severity of their PFO in your data analysis?
So yeah, we do grade it on a scale of 0 to 5, and it's based on the number of bubbles that we count. For this analysis, we didn't look at PFO size.
Okay. The next one I have is actually for Carly as well. Why only men? Do men have a higher prevalence rate overall with PFOs?
No, I don't think so. We just haven't looked at women yet.
Okay. All right. The next one I have is actually for Donna. So with one of your conclusions that you had was that I think it was SSD is not sensitive enough to elicit a response. Is that, would you mind kind of talking a little bit more about that?
Yeah, so we know that, not only our data, but previous data, it says that it's supposed to detect this overall ventilatory response. And so it can do that. But if we're really looking at, if it also can look at the sensitivity, such as if someone has a physiological difference, such as a PFO, can it detect differences between groups in that way? And so when it said, or when I wrote, it may not be sensitive enough, that's in comparison to the alveolar data that says that it is different between groups, but the SSCD isn't showing differences between groups.
Okay. Great. Thank you. Let's see here. Are individuals with sleep apnea protected if they have a PFO?
Protected from even lower PO2s? Certainly not. PFO, if anything, is going to exacerbate any decreases in arterial PO2 as the blood shunted through the PFO isn't experiencing pulmonary gas exchange. So if anything, I would suggest that a PFO is probably going to make sleep apneas worse.
Okay. All right. Good. All right. The next one's for Caitlin. Did you take a closer look at the association between pulmonary vascular resistance and cytokines? And then it's a follow-up to that too. It strikes me that the delay increase in resistance might be compatible with the presumably delayed increase in systemic cytokines. Do you have thoughts on that?
Yeah, so I did look at, I did do a multiple regression with the vascular resistance as well. And there were no significant overall models. There were a couple of the cytokines. I believe IL-18 was a significant variable, but the overall model wasn't significant. So there definitely could be some players there. One of the main limitations is that we don't have blood samples from all of the time points we took the ultrasound measures. And so being able to look at whether there are any associations at each of the individual time points, I think might be a future direction to help kind of answer that question a little better.
Okay, great. Thank you. Thank you. The next one I have is for Carly. You said that it was in a thermal neutral environment. Do you feel the hot environment may have affected overall results that you collected? Do you feel it'd be somewhat similar?
I think it would be interesting to see the effects of the ambient temperature and then them regulating heat dissipation mechanisms at the same time to see whether, like, how heart responds when you're actually getting hot and then you're having to do increase in skin blood flow and stuff like that.
Yeah, yeah.
It would probably
Yeah.
alter some of the responses.
Okay, okay. Good. The next one I have is for Caitlin. Caitlin, so were there any differences in based off the PFO positive and PFO negative individuals' experience from the study based in any feedback they provided? Did you see any differences kind of between the feedback they provided?
Feedback in what sense?
Yeah, Andrew, would you maybe want to follow up with that?
He may not have access. He'll have to write.
Oh, okay. Oh, sorry. Yeah, I'm not exactly
You
sure.
have to type, Andrew, you just need to type.
Yeah.
Sorry about that.
Oh, no worries.
Yeah. Okay. Just any verbal feedback for the two groups, just, the difference between PFO positive or PFO negative.
So, so this was also part of a bigger study that looked at incidences of acute mountain sickness as well, and trying to see if there were differences between PFO negative and positive, to see if those of the PFO got sicker at altitude. So that was part of the rationale behind the initial study. So, and I was not involved in the actual chamber, the chamber portion of this. I did all of the blood analyses. So whether or not there were differences in feedback within the chamber, I can't fully say, but there may have been.
Okay. Yeah. Okay, great. Thank you. So I have exhausted my questions. I think I've gone through all of the questions that I have in the chat here. Are there any other questions that other people have at this time? Okay, not seeing anything pop up here. So we're finishing up a little early here. So I just want to remind everyone that, oh, there we go. For anyone, PFO closures who can. Sorry, I'm just reading one more.
While Charlie's reading that question, just to remind people, for the next session, remember you need to leave this Zoom session, and the next thematic session is the next Zoom link. Okay, so you'll get kicked out of here at the end, but know that the next one will be starting at 4:30 Mountain Time, 3:30 Pacific Time.
Okay. I do have a question that came in here. So, and this is for anybody. So PFO closure is becoming common, especially with large grade 5. Can it be assumed that PFO differences be mitigated by a closure device?
So we've done a few studies on PFO closure. There do seem to be definitely some changes with closure of PFO. We've seen a significant decrease in total pulmonary resistance, as well as significant increases in pulmonary gas exchange efficiency. I think Caitlin might be able to speak to some of the inflammation changes better than I can, but definitely PFO closure has beneficial effects on a lot of these outcomes that we've talked about.
Yeah, for the inflammation side of things, and granted, this is with an N of 3, so not anything big as of yet, but in the 13 cytokines that I showed today, there was a decrease. All 3 participants in this closure study significantly or did decrease their inflammation after the closure of their PFO.
Good. Well, yeah, once again, I've exhausted the questions here. So once again, similar to what James said, we will meet back at 3:30 and I hope everyone can join us for the